In our earlier discussion, we were talking about the concept of well-born or the eugenics given by Francis Galton. As we have discussed, know that he was primarily referring to certain types of behavior that has to do with the genetics and how good traits have been transmitted in you, so that you come forward with the desired pattern of behavior. He was talking about criminal behavior, he talked about alcoholism, immoral forms of behavior. So, let us look at a couple of them. First, let us talk about alcohol intake. We all know that for proper metabolism of alcohol, the enzyme that is needed is aldehyde dehydroxygenase. Now, this enzyme actually regulates the entire metabolism process of alcohol. So, whenever you take an alcohol, your body needs to metabolize it, and for metabolism you need this enzyme. This enzyme in turn is regulated by genes, and variation in the genetic code can also result to inactive enzyme. This primarily would mean that if you have inactive enzyme in you and if you consume alcohol, your body will not be able to metabolize it. Okay? And if you are not able to metabolize it properly, this again would mean that you will turn sick the moment you consume alcohol. Now, those who have been taking alcohol and have inactive enzyme in them, their body will actually not accept it, and because you realize that your body is not accepting alcohol because of the inactive enzyme, you actually will start refraining from taking alcohol. Now, this is how your genetic predisposition will control or will influence your drinking behavior. Because of the inactive enzyme that has to do with gene, that is actually regulated by gene, you do not think of consuming alcohol. Whereas, when you look at the other side of it, you have the active enzyme in you, but then you are a man of religious belief, or you have simply accepted the cultural restrictions to drinking behavior, and therefore, you never feel taking alcohol, you have never taken it. Now, you take the same behavior, what Francis Galton was referring to, that you take alcoholism for example. Now, for alcoholism, when you look at the same behavior from the two perspectives, you realize that in one case you have the inactive enzyme because of genetic reason that leads to refraining from alcohol intake. On the other side, you do not have the genetic control, rather what you have is cultural control, a religious belief that actually moderates your behavior. This is actually environmental influence on drinking behavior. So, the same behavior, alcoholism you are looking at, but then at one end you realize gene controls it, the other end you realize that the environmental influences control it. Let us take another example. We are talking with a reference to the intellectual outcome of an individual. If a pregnant woman consumes alcohol during a specific period of pregnancy, the intelligence of the child is adversely affected. This is what studies have found. So, intelligence of the offspring, the intelligence level, the cognitive capability of the offspring will primarily depend or get influenced by the fact whether the mother consumed alcohol during certain specific period or not. When you look at the other thing, say it has not to do with alcohol, but say you are actually an inhabitant of a house which is painted by lead based paints. Now, lead might again affect adversely your intellectual capabilities, and once again the outcome in the child is intellectual problems, cognitive problems, but then the deficiency that you find in the individual either has to do with alcohol that the mother consumed, or that has to do with the lead that was there in the paint. These are once again the examples how your environmental conditions actually affect development of the brain. Remember that study we were referring to how anatomy of the brain, the neurotransmitters, the interconnections of the neurons, these are all controlled by the genes. Today, we are looking at the fact that fine you could still have environmental determinants that influences it. Take novelty seeking behavior for example, something that has been reported to be correlated to identical twins. Now, studies have confirmed association between novelty seeking behavior and D4DR neuroreceptors. Now, D4DR is a dopamine D4 receptor, and these neurotransmitters also influence mood regulation. So, the type of mood that you experience has to do with this dopamine receptor, and this dopamine receptor also influences the novelty seeking behavior. So, you find that the relationship between the gene expression, the environmental condition and the manifested behavior actually are too complex. Look at this handwriting pattern. You all are aware of the reading disability called dyslexia, where you suddenly find here H has been written in the inverted form. It is a mirror image of the H actually. So is the case with the S, and then look at this full string. The child who has written ABCD or has written the days of the week, you find certain patterns emerging out here, and actually these are some of the prominent indicators of dyslexia. As many of you might not be familiar with dyslexia and we have not discussed it in this course, let us try to understand what actually happens in the cases of dyslexia. You are looking at two hemispheres of the human brain. Language is primarily considered a left hemisphere function. This is the temporal lobe of the left hemisphere. You now see the four lobes: the frontal, parietal, temporal and the occipital lobes. During formal education, we are trained for different reading and writing skills. The three neural systems for reading are located in the left hemisphere of the brain. You see them as regions 1, 2 and 3 on the screen. 1 is in the region of the inferior frontal gyrus, which is also known as Broca's area. 2 is in the parieto-temporal region, and 3 is in the occipito-temporal region, which is also known as visual word form area, that is VWFA. This plays significant role in fluent reading. It has been observed that compared to normal individuals, those with dyslexia have less gray matter in the left parieto-temporal area. Many of them also have less white matter in the same area, and less white matter could adversely affect communication between the brain areas. The interior system in those with dyslexia is slightly overactivated compared to non-impaired readers. In contrast, the two posterior systems are underactivated. The underactivation of the left posterior reading system is the neural signature for dyslexia. We all know that dyslexia is moderately genetic in its origin, and when you try to trace the genetic origin of a dyslexia, you find that phonological awareness is linked to a region on chromosome 6, and phonological awareness is very much needed for developing proper reading habits and to be able to read and write properly. Now, decoding of a single word, it has been found that it is linked to a region on chromosome 15. Now, phonological awareness to chromosome 6, you have chromosome 15 that has to do with decoding of a single word, and both these faculties are actually they play important role in deciding your proficiency in reading and writing. So, these two faculties are actually key to reading disability, and again you find that for phonological awareness as well as for decoding of the single word, you have a genetic control, chromosome 6 and chromosome 15. So, this actually tells you that fine, you have a genetic origin to dyslexia. Now, the specific gene and the mechanism are yet to be understood. The location has been identified. We know that fine, you have two genes. Okay, we have discussed gene chromosome 6 and chromosome 15, certain specific regions, but then the full mechanism is yet to be understood fully. This is just an example of difference in the scientific approach towards understanding of dyslexia. Here you see the pedigree showing family members who were included in the linkage analysis for dyslexia. The pedigree has those members who tested positive on the test of dyslexia, and those also had history of reading problems, as well as those who tested only positive and those only with positive history. This is how the experts of genetics attempt to understand dyslexia. You now find example of neuroimaging studies on dyslexia, where the focus is on the broad anatomical characterization of brain dysfunction. You can clearly see the surface rendering with underactivation in red and overactivation in green. Let us now look at the distribution of human characteristics in any given population. When you look at your surrounding very carefully and observe the distribution of the traits, right in the beginning we saw a long video actually showing you the difference with respect to all types of phenotypic characteristics. You find that there is probably a likelihood that you would come across a curve that is technically called as the normal distribution curve, or in general terms when you say that find you are little close to me, or say x is little close to y. So, basically you realize that there could be a pattern of distribution of certain traits when you look at your environment. So, most of the variables of interest in psychology are continuous, and the continuous probability distribution actually adequately describes such observations. So, when we consider human traits, many of them are not looked in terms of their presence or absence, rather they are actually looked at with respect to their spread in the population. So, for example, if I look at say personality, okay, if I look at say introverts, extroverts, I do not look at the presence or absence of a trait. Okay, you cannot say that no, I have 0 personality, or I do not have personality at all, or you cannot say that I have 0 intelligence. So, when you take psychological traits, you do not talk in terms of the presence or the absence of the trait, rather what you actually focus upon is the distribution, the spread of the given trait in the population, and whenever you look at the spread, you actually come across a curve. So, when you graphically plot it, the population variation is expected to follow this normal distribution, and any single gene can account for only a small proportion of variation in any given trait. The graphical representation of the normal probability curve actually looks like a bell shaped curve. Right now, we will have a look at it, and this bell shaped curves or normal distribution curve assumes that the mean, the median and the mode, they coincide at a single point, and therefore, this curve declines symmetrically in either direction from the maximum point. Let us look at the normal probability curve. 68.2% of the population clusters between 0 and plus minus 1 sigma, that is 34.1% between 0 and plus 1 sigma and 34.1% between 0 and minus 1 sigma. 13.6% between plus 1 and plus 2 sigma, as well as minus 1 and minus 2 sigma, and finally, you have 2.15% between plus 2 and plus 3 sigma, as well as minus 2 and minus 3 sigma. It is important to note that the curve never touches the horizontal axis. Almost all the values of the observation are within the range of mean plus minus 3 sigma. If you add the percentage covered under the curve, it is actually little less than 100%, and this gives scope for exceptional cases. So, you could have an exception, but then this exception actually you realize that this is not covered under the normal probability curve. But by and large, when you look at the spread of traits across the population, you find that you can actually plot a normal probability curve. Now, in this normal probability curve, you have an option. Either you suddenly realize that a particular trait actually swings toward in a particular direction. So, instead of maximally coming between minus 1 and plus 1 sigma, or getting normally distributed, you realize that suddenly it has clustered in a given segment of the normal probability curve, and this is what is called as skewness, okay, that the curve is skewed. So, a particular trait suddenly going in a particular region of the normal probability curve, that could be one possibility. The other possibility could be that a given trait in a given society, you suddenly realize that a major chunk of it comes between minus 1 and plus 1 sigma, and this could be the state when the curve will suddenly the nose of the curve, the tip of the curve will go very high, okay, and the rest of the regions of the normal probability curve will have very minimal people representing the trait, and this state is called the state of kurtosis. You remember right in the beginning, we looked at this image primarily saying that in behavioral sciences what we are more interested in is those desired traits that are needed for the healthy, happy survival, the productive outcome of the individual. Now, the traits that we were actually looking at, personality, memory, critical thinking, all these abilities. So, when we look at it and try to look at the spread of these traits in the given in population, you always realize that you could by and large come across a normal distribution curve, where these traits, all of them, okay, whether it is problem solving ability, whether it is thinking style, whether it is memory, your intelligence, personality, or whichever trait you talk about, okay, all of them can actually be plotted in this normal probability fashion. Now, when we look at these traits, and when we look at the variation that actually exists in the given population, here comes a major challenge from a practical viewpoint. Our educational system actually does not pay attention to this biological variability. So, most of the activities are actually focused as the average population. So, those with problems are considered deviant from the average, and therefore, are not unique, who require things differently, but actually you should be able to think that fine, this is probably a segment of the population, or this person concerned who is falling short on certain parameter in the educational system actually falls on the -2, -3 spread of the normal probability curve, or in some other area. Most of the things are actually designed for those who are in a very average population. So, the moment you start moving at the two ends of the normal probability curve, you realize that the system actually considers you to be a little deviant. Now, one-to-one mapping of the trait and the gene is extremely difficult. Even the gene disease connection is not very straightforward. So, any single trait is an outcome of more than one genetic factor. You remember we have talked about polygenic conditions, where you have the additive effect of more than one gene, and then we have also seen that the genes actually are not sufficient in themselves to get expressed, therefore, they require a conducive environmental situation. So, any given single trait actually cannot be considered as an outcome of a single genetic factor, but there would be more to it, and the interaction of these factors might have different effects depending upon the individual's genotype. Now, the impact of environmental factors also depends on one's genetic makeup, and findings of behavioral genetic studies are often difficult to replicate, something that is usually possible in behavioral sciences. Okay, here we find for behavioral genetics it is extremely difficult to replicate the same observation. Manifestation of a trait of course depends upon the environmental factors, as we have been discussing here, and there can be an interaction effect of the environmental factors which will keep influencing the expression of the genetically determined traits. Hence, a trait might be considered as an outcome of a specific genetic variant, but that might not be sufficient for the trait to get manifested in the behavior. This is an illustration of DNA trait pathway. We always have the DNA, RNA, protein trait, this full pathway that is always looked upon at. So, primarily what in behavioral sciences we are looking at is the reflected trait, and of course, when you start looking at the pathway, when you start tracing them, you find that it is not very straightforward. So, the gene protein trait pathway needs to be understood even for traits that can be identified with a particular gene, because the predictive power of a set of genes contributing to any trait is extremely limited. Genes appropriately express themselves while responding to internally and externally induced stimulation. After proteins are made, certain factors must intercede for the realization of neurons and behaviors. The external agents that we look at in our environment such as pesticides, industrial chemicals, heavy metals, air pollutants, okay, they can actually change the expression of a gene. Let us take for an example how the environmental conditions, how environmental pollution, the things that are those stuffs that are available in our environment, how they can alter it, how they can modify it. Let us take for example, the anti-apoptotic B cell leukemia, or what is called as the lymphoma 2 gene, the BCL2. Now, they are located on chromosome 18. In non-Hodgkin lymphoma, that is the NHL, this BCL2 is translocated to chromosome 14. Now, remember that you find it located on chromosome 18 and then it gets relocated in one state, the NHL condition, on chromosome 14. Researchers, what they have found is that there exists a positive correlation between the NHL and the pesticides, okay, and series of pesticides. You can look at lindane, you can look at fungicides, you can look at toxaphene. Okay. All these pesticides, they have been found to have a positive impact on NHL. Now, what you can infer is that occupational exposure to pesticides can actually increase the frequency of translocation. Okay. So, what we were actually looking at that from chromosome 18, if BCL2 gets translocated to chromosome 14, this could be because of the occupational exposure or environmental exposure to pesticides, and this exposure to pesticides can actually lead to cancer. So, what actually I am trying to make a point here is that although there is no genetic expression of the cancerous cells here, the lymphoma here, the BCL2, is the fact that no translocation leading to cancer is actually an outcome of external environmental influence. Environmental exposure can couple with genetic events such as translocation that we have talked about right now, and can pose a risk for a given disease. Now, several gene regulation mechanisms are susceptible to modifications under environmental influences, and this means that many of the diseases that we were actually considering to be very straightforward in nature, that a particular gene actually causing a particular disease, this mechanism needs to be relooked upon, needs to be understood, because this whole pathway, when we draw this pathway, the DNA, RNA, protein traits, this pathway is not very straightforward. A variety of genotoxic environmental factors, say for example, cadmium, pesticides we have talked about, they can cause DNA strand break or fragmentation. We all know about diabetes. Now, a positive relationship has been found between urinary cadmium and diabetes. This basically suggests the role of cadmium exposure in diabetes etiology. There are examples of genomic reactions and adaptation to environmental stimuli. Lumsden and Wilson came forward with a theory of gene culture coevolution, which basically talks about a very interesting fact suggesting that purely genetic or purely cultural modes of transmission, they are less likely to actually sustain this demand of evolution. So, evolutionarily stable traits would be only those that are actually influenced by both the genetic influences and the cultural influences. If they are put in isolation, probably they will fail this whole demand for adaptation, and therefore, they will not be very significant, very stable from an evolutionary viewpoint. Cultural transmissions largely ensure resemblance between siblings or other members over and above the shared genes. You can take an example. Usually, when you say for example, behave in a particular way, your parents tell you, or those who are close to you, they will tell you, look at your brother. Basically, what they are actually telling you is that even if you have genetic variation as a sibling, look at your brother. This means that your brother has something that you can actually imitate, or rather it is recommended that you should imitate. Say for example, my brother has a habit of having long sitting hours and reading and writing. I do not have this, and this is something that is desirable, and therefore, irrespective of the fact that my brother puts in more effort and I put less, I am told that fine, you should actually imitate your brother. This means that irrespective of my cognitive capability or irrespective of the difference that we genetically are predisposed with, I am supposed to imitate, because my brother has something that is socially desirable. This actually means that the society in general will not actually allow you to go ahead with the variation that you have because of the genetic predisposition. Rather, there are cultural demands that are put on you. Okay. We have right now talked about the alcoholic behavior. Now, intake of alcohol, of course, metabolism is one factor attached to it, that whether you have an active or an inactive enzyme. But then irrespective of the fact that you have an inactive enzyme that refrains you from taking alcohol, you have a socially desired thing, and if the social rule does not allow you to drink, then of course, there is great pressure on you to comply to it. This basically means that irrespective of the fact that I am genetically predisposed to an inactive enzyme, and irrespective of the fact that you are actually having an active enzyme, okay, there is a cultural compulsion that both of us should not consume alcohol. So, this is what I mean to say here, that cultural transmissions actually ensures that there is more and more resemblance. This means that the chances of variations are reduced. I will now like to put a very interesting study in front of you. Ruston et al. Examined the cultural and genetic inheritance of individual differences in monozygotic and the dizygotic twins. It is extremely important for the students of behavioral genetics to understand the concepts associated with monozygotic and dizygotic twins. Twins can either be monozygotic or dizygotic. Monozygotic twins are also called as identical twins. They develop from one zygote and form two embryos. Usually, the identical twins share the same chromosomes. Majority of the monozygotic twins share the same placenta, even though they are into separate amniotic sacs. About 18 to 30% of the monozygotic twins have separate placenta as well as a separate amniotic sac. About 1 to 2% of them share the same placenta as well as the amniotic sac. Dizygotic twins are also called fraternal twins. They develop from two separate eggs fertilized by two separate sperms. Each fraternal twin has its own placenta and amniotic sac. Therefore, all zygotic twins are dichorionic. Dichorionic is a state if twins have separate placenta. Monochorionic is the state when the twins share the placenta, and monochorionic diamniotic is the condition if twins share the amniotic sac. But the reason I am putting this study here is actually the fact that he was looking at cultural and the genetic influence on certain extremely significant social traits: altruism, your tendency to be very helpful to others; empathy, nurturance, aggressiveness and assertiveness. And of course, he was using the statistical technique. He used the maximum likelihood model fitting estimation, and interestingly this is what he came forward with. So, you have additive genetic variation, you have common environmental variation, but then you also have a significant effect of the environmental conditions. Okay. And actually, when you look at these socially significant traits, you realize that the genetic variance and the environmental variance both play very important roles. And therefore, when you talk of gene cultural interaction, when you consider that both of these will significantly affect a trait, then this is true. You cannot look at a trait exclusively with respect to the genetic influence or specifically talking about the environmental influence. There could be another way of looking at it. When you look at the full range of deprivations, either it could be an emotional deprivation, it could be cultural, economic, physical or cognitive in nature. Take for example, you have emotional deprivation. You are not given with the care, concern, compassion that usually others get, and therefore, you are deprived of certain set of emotional experiences. There are say cultural variations, your culture deprive you of certain things, or say study we were talking about the economic deprivation, where you do not have the type of commodities, the type of exposure, the possibility of having something that I have simply because of the economic deprivation, or you are physically deprived of something. You do not have no things in your environment that can actually cognitively stimulate you. You have something that can actually deprive you of cognitive influences. Now, all these deprivations will somewhere influence you, and the influences will be extremely strong in nature. We have already talked about cultural differences with respect to its influence on perception when we were talking about the perceptual processes. Something as small as say naming the colors, for example, you find that there is a cultural difference there, and this difference is based on the physiological difference in color vision. Now, some languages we have already discussed that they do not have certain colors simply because of their geophysical location. For example, some languages is spoken in culture near the equator, they fail naming colors at the blue end of the spectrum. Now, green and blue, blue and black, or green, blue and black are not given different names in these culture simply because they are located in a geophysical condition, and therefore, these colors actually do not exist in their nomenclature, their language, and they do not tell them or they do not identify them differently. Physiologically, these people have increased interocular yellow pigmentation, and this leads to decreased sensitivity for the blue end of the spectrum. Now, you see very interestingly here, simply because you are located on a given place in this world, okay, your proximity with the equator, and then the fact that you do not have these words existing in your language, therefore, you cannot see certain colors in the spectrum. We have also discussed the example of certain visual illusions. The trapezoid illusion condition, we discussed that in the rural Zulu people in South Africa, in Natal, okay, their settings contain very few rectangular objects, and therefore, Zulu language has absence of words for squares and rectangles. When these kids they are asked for the trapezoid window illusion, the rural Zulu children have reported 60% less illusion compared to their urban counterparts. So, this basically means that you have certain geometric shapes available in your environment, you have these words in the language that represents these geometric shapes, and therefore, you are able to identify them. If you do not have these geometric shapes available in your environment, and therefore, you also do not have the words representing them, okay, you will actually not be able to have the illusion. So, this is an interesting influence, how culture starts deeply influencing your perception or your possibility of having a visual illusion. Behavioral genetics, of course, also talks about the importance of the learning of the social roles, where you are actually supposed to acquire, you are supposed to play a role that your society decides for you. For example, you are told to play a very specific role, say as a son or as a daughter. You are the same person is supposed to play another set of roles when you play the role of a father or a mother or brother or a sister, or when you play a different professional role. All these roles, the desired code of conduct comes from the society, and irrespective of the fact that you could have genetic variation, you are supposed to comply to these social norms. So, you realize that there are strong social and cultural influences on the behavior. The trait gets extremely controlled by all these types of learning of the social role. Similarly, we have a whole lot set of the socially desired skills, and these socially desired skills once again you are supposed to acquire them. When you acquire these social skills, actually once again you are supposed to go beyond the limitations that has been imposed by the chromosomal predisposition that you have, and therefore, you are always supposed to go beyond the limitations. Now, the moment you go beyond the limitation set by the genetic code, this means that you have a heavy influence of the environmental conditions. Now, what we have discussed till now, if you recapitulate all of them and you just summate it and try to understand, what we realize is that genes and environment cooperate in the construction of any given organism. Genes, they determine our traits, but then genes require environmental and behavioral inputs to function in a normal course. So, you need the genes to determine the traits, and then you actually also need a conducive environmental conditions. You also need proactive behavioral inputs, so that the genetic expression can actually reach its maximum potential. This is now time for us to recapitulate whatever we have discussed about the genetic predispositions and environmental determinants of human behavior.